Okay, so last section, 17.1, we talked all about just the whole entire idea of gene to protein and where this idea came from and why it's important. Why DNA is important is because we're turning them into proteins, and proteins control everything inside of our bodies, which we've talked about a lot. So 17.2 is about the first step in gene expression or protein synthesis, which is transcription. So this is where we are turning our DNA into an RNA template, we're using the DNA template to make an RNA molecule. So that's what this whole section is about. So just like how in replication we use DNA polymerase to add on DNA nucleotides, in transcription we're going to use a molecule called RNA polymerase because we're going to be building an RNA molecule. And so it does kind of the same job in theory. It adds on RNA nucleotides to a growing RNA strand, just like DNA polymerase did the same thing but with DNA nucleotides. And so instead of needing all those other enzymes like we did in replication, RNA polymerase does it all. It comes in, it unwinds the DNA strand, and it separates it, and it starts building. The good thing about RNA polymerase or transcription is that we only need to free about 10 to 20 nucleotides at a time, whereas in replication we separated both strands. They were never going to go back together of the DNA molecule. Whereas in transcription, the DNA molecule is going to go right back together when we're done. We're just opening it a little bit to take a look at it and closing it again. So that's why we don't need a helicase or anything like that. RNA polymerase can do it all. And basically, do I have a picture? No. So RNA polymerase brings in nucleotides to match the DNA template strand, just like DNA polymerase did in replication. A matches with T, C matches with G, except in RNA, there is no thymine. There's only uracil instead of thymine. So every time there's going to be an A, its complementary nucleotide is uracil instead of thymine. So that's one of the differences. But basically, we're building an RNA molecule that's complementary to our gene of interest on the DNA strand. And RNA polymerase needs to know where to begin, right? In replication, we had the origin of replication, but here we're looking for a very specific sequence on the DNA molecule because we want to build a specific gene. And so what happens is the RNA polymerase attaches to a region called a promoter, which is a specific DNA sequence that says, hey, this is where the gene starts. And then in bacteria, what happens is the DNA, there's something called the terminator, which is what tells the RNA polymerase to stop transcribing. That's not found in eukaryotic cells, but we're going to talk about how they terminate that as well. And the stretch of DNA that we're actually going to turn into RNA that gets transcribed is called a transcription unit. So that unit right there is going to be transcribed. So here is this picture is on page 322, and there's three main stages of transcription: initiation, elongation, and termination. So we're going to start our transcription process, we're going to build our RNA molecule and elongate it, and then we're going to end the process with termination. So those are the three steps. So here is the promoter on the DNA strand. It's a certain sequence of DNA that says, hey, this is where the gene starts, come here, RNA polymerase. This right here is the transcription unit. So this is what is actually getting transcribed. And you can see we have our double-stranded DNA molecule, 5' to 3', 5' to 3'. So the first step is initiation. So RNA polymerase comes in and it separates the two DNA strands just a little bit. See, it's not the whole thing, it's just right here. And it starts building onto the template strand of DNA. And so this right here, the bottom one, is the one that RNA polymerase is using to build a complementary strand. So this is our template strand. And the other strand is still there, it doesn't go away, and that's the non-template strand of DNA. So the DNA is unwound by RNA polymerase, and then it starts building an RNA transcript. Now, RNA polymerase is the same as DNA polymerase in that it can only add on to the 3' end of a molecule. So it's going to go 5' to 3', just like DNA polymerase did. So it's going to starts building 5', and it's adding on to the growing 3' end. And so see how as we move on during our elongation phase, when we're adding on the RNA nucleotides, the DNA goes back together. See how it was open here, but down here it's closed? So RNA polymerase only opens it just a little bit to get to the template strand and then closes it back up again. It doesn't need it to be open the whole entire time. And basically RNA polymerase just moves along this molecule, and it just keeps bringing in RNA nucleotides that match the complementary template strand, and it builds a growing molecule. So the RNA transcript, the RNA molecule, is kind of just like hanging off of the DNA template. And the DNA template is unchanged. So RNA polymerase keeps going. So RNA polymerase keeps going, and it keeps adding on. So it brings in the nucleotides that are complementary to this template parent strand right here. So look, we're not even using the top strand, just the bottom one. And it's adding on to the 3' end, just like DNA polymerase did. And then when it's done, the RNA polymerase realizes that it doesn't need, that it's done, and it frees itself from the template strand. The template strand is unchanged. And we have our finished RNA transcript, which has directionality just like DNA, 5' end and 3' end. And we're going to talk a lot about upstream and downstream when we talk about these things. And what that means is the direction on the template strand. So the direction of transcription like this is downstream. So we're moving from the beginning. This is upstream down here, and this is downstream at the end. So sometimes we'll say, okay, well, that region is this far upstream, or that region is this far downstream. And that's what we're referring to. It's the direction of transcription. So that was the overview. Now let's talk about it. There's 3 stages, just like I said: initiation, elongation, and termination. And so we need this promoter region, like I was talking about, for RNA polymerase to know where to start. And so it's usually, it's a section of DNA, and it's several dozen nucleotide pairs. And it has a very specific start point in it. So what's our picture? So here's our whole promoter right here. It's a screen region of DNA. This right here is the start point. That is where RNA polymerase is going to start transcribing. But if you'll notice, the promoter starts much further before that, like several dozen base pairs before that. That's because we need this information to call the RNA polymerase there. If we wanted it to start right here, RNA polymerase would need to jump back further. So we just need this extra space to let the RNA polymerase know that's where it needs to bind. And in bacteria, the RNA polymerase just sees the promoter and starts. But in eukaryotic cells, it's not that easy, and we need several things called transcription factors. Transcription factors are different proteins that help RNA polymerase bind to the promoter region and start transcription. In eukaryotic cells, it does not start without transcription factors. And so once we have pulled in all the transcription factors, pulled in RNA polymerase, and we're ready to start transcribing, it's called the transcription initiation complex. So the transcription initiation complex is made up of the promoter region, the transcription factors, and RNA polymerase. Once all those things are there, we can start transcription. So we need all those things there, forming this complex to initiate transcription. And there's one very important promoter in humans, or I'm sorry, in eukaryotic cells called the TATA box. And let's look at that. So here is the promoter. This right here is the TATA box. And so that's just a very specific promoter that is used in eukaryotic cells. And so, again, the promoter is a bunch of DNA nucleotides that call the RNA polymerase, like say, this is where you need to start. And then what happens is several transcription factors will bind. So these are different proteins that are going to help RNA polymerase bind to the promoter region. So we need the transcription factors to start transcription in a eukaryotic cell. So the transcription factors right here, they call RNA polymerase over. RNA polymerase 2 in eukaryotic cells binds to the promoter and to the transcription factors, and then can start transcription at the start point. See how it doesn't start back here where the promoter starts? It starts at the start point right here. And all of this right here, RNA polymerase 2, the transcription factors, and the promoter is called the transcription initiation complex. So once all that is there and ready to go, then we can actually start making our molecule. So this picture right here is everything that's involved in initiation of transcription, the first step. Then we get to elongation. This is very similar to DNA replication. Basically, RNA polymerase moves along the DNA, and it's looking at the template strand of DNA. Remember, there's two, but the bottom one is the template strand right here, according to this picture. So it's going to transcribe the bottom one, not the top one. And it opens the DNA and untwists it and moves along, and as it goes, it seals the DNA back up. Remember that it doesn't leave it open like we did in replication. It closes it back up, and it's open about 10 to 20 base pairs at a time, and then it's closed again. And so it moves along, and it brings in RNA nucleotides that are complementary to the DNA nucleotides and builds a growing RNA strand, and it can do this at about 40 base pairs a second, which is pretty fast. So it's building these growing strands, and a cool thing about RNA is that since it's leaving the DNA template unchanged, is we can transcribe the same gene simultaneously over and over again. So we start, so here's our template strand. We start an RNA polymerase here, it goes, it goes, it goes. As it's right here, another one can start at the same time. So we can transcribe the same gene over and over again, which is pretty cool. And remember that we're adding on to the 3' end of the growing RNA molecule, just like we did with the replication with the DNA molecules. So basically, here's a picture. This is our RNA polymerase, this little triangular shaped thing, and it's on the DNA strand. And remember, DNA is a double helix, so it's unwinding, and as it goes too, it's making sure it doesn't get tangled. So RNA polymerase does the job of a lot of different enzymes that we learned about in replication. It can do it all. So it unwinds the strand, and then see here we have an available T, so it brings in an A and it attaches it to the growing strand. We have an available A, so it brings in a U because in RNA we don't have T's, we have uracil instead. And so it keeps adding and adding and adding, and this is the 3' end right here, so it keeps adding on to the 3' end. And then as it goes, it's going to seal this DNA back up and keep going and leaving it unchanged. So this is the direction of transcription. So up here, this is upstream from where RNA polymerase is, and back here, this is downstream from RNA polymerase, and up here is upstream. So we're moving downstream as we move down the DNA template. Okay, so we just did initiation and or elongation. The last step is termination. So it's very different in bacteria and eukaryotic cells. And your book doesn't go into a lot of detail. Basically, with bacteria, the polymerase gets to a spot on the template strand, and it transcribes it. And so as soon as it realizes that on the RNA molecule, it just built this promoter, the RNA polymerase leaves and transcription is done. So in bacteria, the terminator is transcribed. So the terminator is on the RNA molecule. It's actually the RNA molecule, not the DNA molecule, that tells the bacterial RNA polymerase to be done. And then the RNA is finished in bacteria cells. There's no RNA processing in bacteria. This is different than in eukaryotic cells. Basically, there's like one paragraph about this in your book, and it's pretty complicated, but RNA polymerase gets to what's called a polyadenylation signal, and it's a very specific signal of bases, which is AAA, and it tells basically once it gets to that section, then the RNA polymerase is like, okay, we're finishing up, and 10 to 35 nucleotides downstream, it cuts it loose and the RNA is finished. So it's kind of like a red light. Like if you're driving and you see a red light, you're not going to stop when you get to the line. You're going to start stopping way ahead of time. That way, by the time you get to the line, you're stopped. So it's kind of like that. It's like this signal that says, okay, you're almost done, so start slowing down, and by a couple of base pairs, 10 to 35 later, it's done, cuts it loose, and then we have our pre-mRNA. What this means is that it's a RNA molecule, but it's not quite ready to go into translation yet, so it needs to go through mRNA processing, which is the next step in eukaryotic cells.